Dharamveer, who is the president of SPSTI, to formally extend a welcome.
Professor Javed Agrawalaji, Professor Grover, distinguished professors, scientists, and students. It's really a pleasure to welcome you all to the launch of new series of 12 lectures today. We have received a modest grant for holding 12 lectures. And the grant came from the Department of Science and Technology, Chandigarh Administration. So this grant would cover the lectures on Nobel Prizes, Nobel Prizes, Turing Prize, World Food Award, and Bhatnagar Awards. So this would actually bring a lot of discipline among us to organize lectures on a regular and a systematic manner. So the Nobel Prizes 23 were announced recently between October 2nd and 9th. Accordingly, we started preparing a calendar of lectures. The series starts today with the lecture on Nobel Prize in Physiology and Medicine, and speaker is well-known scientist, Professor Javed Agrawalaji. I welcome him, and eminent scientist and senior faculty of IIT Ropar. We are grateful that he has accepted our invitation. I also welcome Professor Prince Sharma, Department of Microbiology, Punjab University, for accepting our invitation and request to be guest of honor. We'll try to complete these lectures, 12 lectures, during the current financial year, for which we have the grants. Thanks to Professor Grover and my wife, Professor Dharamveer, for arranging and coordinating these lectures. The SPSA is ever grateful to Punjab administration for making its infrastructure available for hosting the lectures. SPSA, I would appreciate for your feedback and suggestions for making improvements wherever required. At the end, I would like to say that this forum is open to every one of you, students, teachers, faculty, researchers. We are absolutely open to suggestions, taking any other initiative which you think is relevant for promotion, popularization of science and technology, not only among us people, but also students and teaching faculty. We have been collaborating with many other institutions and people of the scientists, professors working in the universities located in Haryana. And we have also been getting generous support from the faculty of Punjab University for more than 1.5 decades since we formed the society. So it is because of the love and affection that you have been showing to us over such a long period. The society has been able to establish itself and has emerged as a major catalyst in taking science to the people. Thank you very much, Jai.
I now request Professor Arun Grover, our Vice President of the Society, to give a brief on the lecture series. Professor Grover.
Good afternoon, all of you. I am indeed very happy to have this opportunity to briefly tell you about this series. This year's annual series is the fourth year in a row that we are running this series. But during last 3 years, this series has run in an online mode. But from this year, Dharamveer said that no, we must do it in a hybrid mode. So I said, okay, we shall do it in a hybrid mode. But once you do it in a hybrid mode, we have to find a host for every lecture. So idea is that we will have this series at different departments or institutions belonging to the CRICSI during this year. So we have 6 Nobel Prize lectures. Okay, medicine and physiology, 1, physics, chemistry, then economics, then literature and peace. Added to it is the Nobel Prize in mathematics, Turing Award in computer sciences, and World Food Prize. So the Nobel Prizes are announced October, Nobel Prizes and Turing Awards get announced in March, and World Food Prize happens in the month of May. So he proposed to cover the first 6 lectures by second week of December. Okay, and do all the 9 lectures by March, because if we don't do it by March, then we'll have a backlog accumulated because new awards will come in. So Dharamveer said 12 lectures we have a grant, but we are going to do more than 12 lectures. Because to add to these 9 lectures, every year we have been commemorating National Science Day, which happens in February 28. Then we commemorate National Technology Day, which happens on May 11 every year. Then we commemorate Teachers' Day. We have had Teachers' Day lecture already in the very building. And the speaker is here with us. Sandeep spoke on the Teachers' Day 2023. And then National Mathematics Day is very dear to the president of SPSTI because of his mathematics past and passion. So these at least these 4 have to be added. But then, CRICSI has lots of technology institutes and lots of university departments, and so the technology institutes they commemorate every year the Engineers' Day. That is on September 15th. Then the Department of Statistics and statistics interested people, they commemorate the National Statistics Day. That is Mahanalobby's birthday on June 29th. Added to it is, for the young people, Kalam's birthday is important, and that is October 15th. Okay, then for educational institution, National Education Day is equally important, and there's Maulana Azad's birthday on November 11th. So, okay. So we have already exceeded beyond 12. Added to it is a annual event which has gone on in Chandigarh since the year 2007. That is the Chandigarh Science Congress. We just commemorated the Chandigarh Science Congress, and coordinator of this year's ChessCon is with us as well. So that's a 3-day event. Okay, so this is a minimum configuration. Okay, so, and my guess is this will happen every year. So we ought to have an annual calendar. And so the SPSTI and the CRICSI, the older organizations, must join hands together and prepare an annual calendar, and it should be widely circulated, advertised. And the different CRICSI institutions should consult this calendar while fixing up their convocation dates and the days of their Foundation Day lectures. Every institute has a Foundation Day lectures because these things are all public events. Foundation Day lectures of PGI is a public event. PU Foundation Day is a public event. So are these lectures also public events. So the public events organized by the academicians for the civic society, there should be a platform. The media should also know it in advance. And I think the Chandigarh media, there are so many Chandigarh newspapers and so many correspondents which walk around our campuses. They must also have it in their Google Calendar that this is the annual event they could cover. So from this year onwards, I think we must prepare it, have it widely circulated. Yeah, so that is how I got into it, because Professor Pasi, he was a founder, vice president of SPSTI. When his health became a little shaky, he wrote me in and said, "आप ये करिए, मैंने इतने साल किया है, आप ये करिए." So it's a noble cause. I accepted it, and I'm very happy doing it. Okay, and as far as possible, I try to be physically a part of this every year. But we should make it in a manner that more people are part of it. Already the members of the Chandigarh chapters of the science academies, and we have 6 science academies, and there are over 100 members of the science academy serving and retired, living in the Tri-City. We have 3 science academies, then there is a National Academy of Medical Sciences. PGI has over 70, 80 doctors who are members of National Academy of Medical Sciences, serving and retired. Added to it is the National Academy of Agricultural Sciences, then Indian National Academy of Engineering. So minimum 6 science academies are there, and nowadays we also have a forum of retired vice chancellors and retired directors. So the audience is large. So doing it in a hybrid mode and online mode is good. More people can attend to it. And added to it is that the technology and pandemic has made enhanced the bandwidth and enhanced the archives of YouTubes and this and that. So SPSTI maintains recordings of all the lectures. So every lecture is available. You can, if you miss it or you want to listen to it, who got Nobel Prize for what 3 years ago, you can of course find it on the internet, but you can also go to the SPSTI site and you can get all these details. Advantage of SPSTI site is that speaker can be consulted. Typically it is someone amongst us. So the speaker can be posed questions. If you want to get that lecture repeated in any college, university, this, that, same person, the lecture is already ready with him or her. So you can ask him or her to give that lecture. So it's good for the department, for everyone. So now this CRICSI stands expanded into a cluster for the northern Indian states. CRICSI never had any budget of its own, but this cluster has a budget of its own. So since its budget has a budget of its own for first 3 years, it is going to be converted into a Section 8 company, so it should have budget forever. So people who are going to look after this cluster, I think they should also assume responsibility that a better web page would be created, which would remain updated, interactive, and so on and so forth. So it's a good thing to be a part of, and I hope doing these lectures in a hybrid mode at different locations, for which we already have a grant. If we do 12 lectures, so at least 12 audiences in different locations will become alive to this act of ours. So with this, I have already taken it too long, and you have all not come here to listen to me. You have all come here to listen to an expository lecture on Nobel Prizes by a very eminent scientist from our region, Professor Javed Agrewala, who is a Patnagar awardee and also a winner of so many other awards. Thank you very much.
So welcome, Dr. Pooja. We are so glad that you could come and join us. Please take it. Thank you so much, Professor Grover, for bringing us up to date on the series of lectures. And now I request Dr. Jaspreet Kaur from Punjab University. Jaspreet is from Botany Department and is very active in outreach work on behalf of CRICSI. Jaspreet will introduce the guest of honor today. Jaspreet.
Thank you, ma'am. It's an honor to introduce Professor Prince Sharma, former Dean, Faculty of Sciences, Department of Microbiology, Punjab University, Chandigarh. Professor Sharma is working in the areas of reverse vaccinology, antimicrobial resistance, industrial enzymes and biosensors for microbial diagnostics. He has published 119 research papers, completed 14 research projects funded by DBT, DST, UGC, ICMR, DFS, and guided 29 PhD students. He has received GlaxoSmithKline Vaccine Award for his work on reverse vaccinology in ECCMID at Amsterdam. His research paper on reverse vaccinology in scientific reports was selected in top 10 papers in 2017. He has been conferred with the Srimati Prem Lata and Professor D.V.S. Jain Research Foundation's Best Research Publication Award. He is selected amongst the top 2% scientists in the world by Stanford University in the year 2022. Defense Threat Reduction Agency USA sponsored him for the European Biosafety Association Conference at Madrid 2017. He is a Sandia National Laboratory certified trainer at Punjab University for biorisk management and has conducted several training workshops for the same. He is one of the highest money grosser for Punjab University through technology transfers and consultancy. He has 2 patents granted and 2 enzyme technologies transferred to industries like Cadila and New England Biolabs USA. He is an advisor to AI Novo Biotech USA and consultant to Trish Veda Naturals. His current research work is in collaboration with L'Oreal Paris, Panacea Biotech, and AI Novo Biotech USA. He is a fellow of the Academy of Microbial Sciences, India. So I now invite Professor Sharma to kindly enlighten us with a few words of wisdom. Thank you.
Thank you, Jaspreet. Professor Grover, Dharamveer ji, Professor Kia Dharamveer. I'm really feeling great actually, and very thankful to all the 3 of you and the other organizers of this SPSTI programs and this series. Not just this series, but all other series which you have initiated, and you have been doing this lecture series for a long time. My thanks are from the whole of the scientific community to you, that you are promoting such a noble cause and such a noble thing, this science caliber, scientific temper among the researchers in at least this part of or this part of the region of India. SPSTI, I really believe, is doing a great job in this spread of scientific knowledge, and I believe that all the students of microbiology department and other departments who are gathered here today, they will be very much benefited. And this particular new series, which is being initiated today and will be finished within a span of about a month or so, again, this is a very commendable thing. And to think about this, why not have a series on Nobel laureates, Nobel work? And the very first one which you have selected is on the field of physiology and medicine. Of course, very close to us because we are in this field, and who can forget the role of Catalin and Drew Weissman in the development of such good Pfizer and Moderna mRNA-based vaccines. It's because of their work on mRNA, the nuclear modifications in the mRNA, that we have seen such good protective vaccines um, from Pfizer and from Moderna. And it's these vaccines which have been able to protect us from this COVID-19 pandemic in a very big way, actually. There have been other vaccines, but these were probably treated as one of the best vaccines as far as their protective efficacy beyond 80-90%, which goes by that, actually. And it's probably not the end, because as many people as microbiologists believe that maybe this COVID-19 was not the last pandemic. There may be more in the, God forbid, but we can have more of the diseases, there more of the research, more of such kind of noble works they are needing. And as a microbiologist, as Dr. Agrawala will agree with me, that we are already going through a silent pandemic of antimicrobial resistance, where microbes have become so resistant to antibiotics, and we are very soon we will be out of all the antibiotics which will be working against the microorganisms. Dr. Agrawala is a very well-known scientist working on microbacterium and finding different kind of vaccines against mycovaccine.
धंधा का काम करा
We in our lab also, along with in collaboration with Panisha Biotech, we are trying to develop vaccine against the escape group of very highly drug-resistant Bacteromonas, which have been the cause of many deaths now because antibiotics are not working against them. So it is
नहीं डैडी
in this
उसे
light
इलाज चलता
that
है ना
more of such kind of research is needed, where people like this Hungarian scientist Catalin and Drew Weissman, they are needed. More of such vaccines are needed, and scientific temper, I believe, is further much more needed because this is the one which keeps you keep doing your research. How many people know that this researcher Catalin, she was demoted by University of Pennsylvania. Her salary was cut. She was not found fit for the faculty not long time back, and now today University of Pennsylvania is celebrating her Nobel Prize. I believe that okay, that is what science is. That don't get deterred by what a few people think. Ultimately, it's your work which is going to speak volumes about this thing. And we really, this is and really an example actually. This work that okay, even if you are down but you are not out, even if you have been severely punished by a university, but still you have come out with vengeance and you have won a Nobel Prize actually. And today's lecture is very apt and very suitable. Boy has been selected to talk about that lecture. So, old friend of mine, we have been together since we were. I was assistant professor and he was a scientist in. He just joined and we were living in the same street, same walla actually. So since then we know each other and I know he's done great actually. But Nagar award and this one, he has a lot many awards actually. So I really believe that SPSTI has done a great job in selecting him and finding this person to talk on this particular topic. And I feel that he's going to tell us very much about this Nobel Prize, and more of the Nobel Prizes will follow actually. So great, Professor Dhambir, Dhambir ji, Arun Roji, and we have others organizers of SPSCI. Great that you have organized, and microbiology department is again thankful for here for our students,
ये दो
and
छोटे
this will
छोटे
be
स्पून
great for our students. Thank you so much, please.
समोसे में मिल गए प्रांजल
Thank you, Professor Prince Sharma, for those encouraging
चलो
words. And now I request Dr. Pooja Devi to introduce today's speaker. Pooja is from CSIO, a very distinguished scientist herself, and she is a member of the INIAS, Indian National Young Academy of Science. Pooja.
A very good afternoon to one and all. First of all, thank you for the opportunity. It's an honor and privilege for me to be here to represent INIAS, who is jointly organizing this event. 2 weeks back, I was in US only. I was in Philadelphia, where UPenn, the very place Professor Karikad, Professor Wiseman was, and I was there when this Nobel Prize was announced. So I witnessed the celebration actually that happened in Philadelphia in the UPenn and had an opportunity to meet both of them. So that's why I took a chance to come specially for this lecture, and today we are going to listen about this particular discovery. So I'm going to introduce our speaker, who is eminent in his own field, Dr. Javed Agarwala. Dr. Javed Agarwala completed his PhD from Agra University, Agra, and joined a faculty position at CSIR Tech in 1989. He has been a visiting faculty at Hammersmith Institute, London, and Trudeau Institute, USA, and
तो
he
फिर क्या
joined
करना
Indian Institute of Technology, Ropar, in March 2018. Dr. Agarwala Group has made seminal contribution in the area of immunology of infectious disease and vaccines, as evidenced by his publication in reputed journals, international patents, technologies, and high citations. He has been the recipient of several awards and recognitions, including prestigious Shanti Swarup Bhatnagar Award, J.C. Bose Fellowship, National Bioscience Award for Career Development, UP Science Award and endorsement as a fellow of all three national science academies, including INSA, NASI, and
हाँ
Indian
जी
Academy of Science.
ऐसे ही
So, with
खत्म
this brief introduction, I take this
हाँ जी
opportunity to invite
क्या हाल
him
है
for his talk.
कि हाँ दिल्ली गया है आज ही हम तो ठीक हो
So good afternoon to everybody. So
आ गया
I'm
बाबा
highly grateful to
डेटा
Professor
साइंटिस्ट भी नहीं
Grover and the organizers for giving me this opportunity to deliver this talk. And it's always been a pleasure to be here at Punjab University. Punjab University is like more of like a home institute since I've been visiting
दो
Punjab
तरह
University
फोन है
since 1989.
कितने चल रहा है सही
So today
नेट नहीं सही चल
I'll be talking about
रहा चक ले जा
this important
दोबारा ਐਡਾ ਕੋਈ ਵਧੀਆ ਲੈਕਚਰ ਲੱਗ ਨਹੀਂ ਰਿਹਾ ਮੈਨੂੰ ਬੋਰਿੰਗ ਜਿਹਾ ਲੱਗਦਾ ਮੇਰੇ ਕੋਲ ਨਹੀਂ ਸਮਝ ਆਉਂਦੀ
prize of physiology and
ਸੇਵ ਕਰ ਲਾ ਨਾ ਫਿਰ ਬਾਅਦ ਚ ਸੇਵ ਨਹੀਂ ਨਾ ਹੋਣਾ
in
ਵੀਡੀਓ
2023. So I hope this is also my slides are visible to people online as well. Can you just check whether it is visible?
it's not visible
Zoom पे तो शेयर की सारे फोन में नहीं चल रहा पीपीटी को मिनिमाइज कर दो थोड़ा सा खत्म हो
As a share me.
स्क्रीन शेयर को ऑप्शन का स्क्रीन शेयर का ऑप्शन नहीं दिखा रहा स्क्रीन शेयर का ऑप्शन नहीं आ रहा कहीं और होगा
आप खुद दीजिए सर ये खुला नहीं ये ये भी नहीं है सर आई नहीं ये ब्राउजर से है ना रीजॉइन करना पड़ेगा पोस्ट बनाया सर रीजॉइन करना पड़ेगा ये किसी फोन ब्राउजर है।
So, okay.
दोबारा से सर लीव करके आप दोबारा से लीव मीटिंग करिए सर। ये सर का
There seems something wrong with the audio. Are you trying to correct the problem or it is not possible? ये इसके ऊपर ऑनलाइन प्रॉब्लम है कोई लगता है मुझे, अगर मेरी आवाज आपको आ रही है तो।
Yes, sir. Yes, sir. Sir, give me 5 minutes only, sir. Please.
तो इसको बंद कर दें फिर कि
Yes, sir. I'm ready to do it, please.
थोड़ी देर वेट करें?
Sir, only 5 minutes, sir. Only 5 minutes, please.
आपकी आवाज भी साफ नहीं आ रही मुझे।
अस्सलाम वालेकुम वरहमतुल्लाह। कैफ हाल? अल्हम्दुलिल्लाह। For? No, I don't know, because once they prepare the memo, they will get the signature of all the attendees, and then I think they will send to the His Excellency. So far, no news.
ममून जी आपकी
Inshallah.
कर दीजिए। ममून जी आप इसको
In
म्यूट
your
कर।
office, are you free for a few minutes? Yes? Okay.
डॉक्टर ममून इसको म्यूट कर लीजिए अपनी साइड से।
Very good, very good. Very good, very good. Very good. ठीक है, ठीक है, ठीक है, ठीक है, very good.
ये हाइब्रिड मोड में प्रॉब्लम है।
नहीं सर, लाइव में तो नहीं होता।
अच्छा, Okay, sorry for that delay. Today's lecture, as I told you, it is about the Nobel Prize in Physiology Medicine 2023. And made this lecture in such a way that I realized since it's in Punjab University, so it will be in hybrid of faculty as well as students. So before I go to my main topic on the Nobel Prize for 2023, I would like to tell something about vaccines. So this lecture is dedicated today to Kathleen Telfo and Drew Westman. And let me tell you what vaccine is. So vaccine is nothing. Vaccine is only, an infectious agent which ultimately, is responsible for any disease. So the vaccine, as the word it suggests, vaccine, it is originated from. Okay. So vaccine, as you know, it is driven from Latin word which is called as vacca, cow. And so, as I told you, that vaccine is a biological preparation that improves immunity to a particular disease. So, whatever we get infection, whether the infection is through bacteria, virus, or whatever microorganisms, ultimately we have to take this microorganism and make it into such a preparation that it loses its virulence. So once it loses its virulence, and but it should at the same time impart immunity. Okay, so typically what vaccine does, it resembles a disease-causing microorganisms. And this is an agent which ultimately stimulates our body, it primes the body, and it not only protects, but it also instructs our body to remember the same agent or same organism for future as well. So in future encounters, whenever we face this pathogen, so ultimately the immune system should remember that this is something which has inflicted a disease, inflicted a pain. So the immune system ultimately going to attack and then remove or react more vigorously against that agent, and it removes. So if I give analogy to vaccine, it is something like a helmet. So in case a person is not wearing helmet, if you see the first picture of a top, if the person is not wearing helmet, so when the person has any accident, what is going to happen? The person is going to be injured. But if the person is wearing, I think you have to stop there. So the person is wearing a helmet. If he encounters an accident, the person will be protected. He will remain, as he is, like, right? So something like this person who still, looks quite well wearing goggles, since he uses the helmet. So it's a protective device, something that you protect yourself before you encounter anything. So vaccine is, different from drugs. Drugs are used to treat us when we become sick, but vaccine is a prophylactic measure. It will not allow you to, feel sick. So if you consider vaccine are among the 20th century most successful and cost-effective public health tools for preventing disease, disability, and death. So it's not only that we are protected through vaccine diseases, but also disabilities, death. So the important part is that we feel that if we get vaccinated, we are protected. No, vaccine of course protects the individual from disease, but at the same time, the vaccine also protects. So vaccine, it protects individual from disease, but at the same time, it protects the entire community by reducing the spread of the infection. Because person will not be infected. Person is going to protect the entire community, entire population, because the spread of the disease, the vaccine routinely remembered, they are routinely recommended for children in preventing early disabilities. So many disabilities which children used to face, like in case of smallpox, in case of measles, polio, so these children, they were prevented. Okay, so let me first go now to history of vaccines. So I consider this person who is historian, Thucydides. This person has a very sparkling talent, and he writes in his book that what he observed after Peloponnesian War, and usually after the war, many diseases were spread. And he observed during Peloponnesian War that people who could nurse the sick, so those people could only nurse the sick who recovered from the disease. So people had disease, people had plague, and these people recovered, and these were the only people who could nurse the disease people. So what does it mean? That the people who got infected and recovered, properly they got immunity. They got natural vaccine. So vaccine, the immunity you can get through injecting vaccines, and at the same time you get immunity through natural infection as well. So the important thing is over here is that continuously when we breathe millions of microorganisms with us. And regularly we are taking these microorganisms. And these microorganisms, we consider that they are beneficial and they are harmful as well. But they are very beneficial since they become beneficial to us, or they become non-harmful, non-infectious to us because we have been immunized with this through natural infection. On subsequent infection, we don't get disease, or we get a mild disease, and ultimately we recover and we are immunized. So it is considered that the people who live in more clean environment, they are more prone to disease, right? So that is why, some people say that people who live in more clean environment, जिन बच्चों को ज्यादा ख्याल रखा जाता है, जितने मालदार बच्चे होते हैं, मालदार पेरेंट्स के बच्चे होते हैं, उनको ज्यादा डिजीज होती है. और जिनके पेरेंट्स don't care about the children, usually very rarely they feel ill. They have got very robust immunity, right? So they face, and this is very evident from the fact that people who live in Western environment, more clean environment, when they travel to, our country or third world countries, developing countries, they have to take a lot of care, or they suffer from diseases. But when we travel any part of the world, we don't, and we are quite healthy. Once I had a collaborator from Royal College Medical School, London. So he visited us in Chandigarh, and he was his family, his wife was also traveling. And first he wanted to see Madurai, Tamil University, because he had earlier some collaborator, some student over there. So when he came back from, he visited, like Bombay, he visited, then he came to Delhi, then he came to Chandigarh. So that's when and Uriah has spoiled your entire country. From Madurai, Chandigarh. So that is when he arrived over here, they took precautions, but then their tummy was upset. Right. So we have more strong immunity. The reason is जो बाहर गोलगप्पे खाते हैं सोई वैक्सीन लगाए और जो बाहर चाट खाते हैं जिसमें मक्खियां बैठी हुई हैं उसमें भी वैक्सीन राइट तो जब मैस में हम खाना खाते हैं उसमें भी मक्खी उसमें भी हमारा वैक्सीन An important thing is that the water we drink, right, if we drink water through tap, it will have vaccine. Right, so the water we drink from tap, we have vaccine. The water we drink from distillery or mineral water, they have pesticides, and you are more prone to cancer. जो नल के पानी में अगर थोड़ा बहुत इन्फेक्शन होगा उसका ट्रीटमेंट है इस पानी में जो चीज है वो अगर हो गया उसका ट्रीटमेंट बहुत मुश्किल से कैंसर अगर हो गया इन्फेक्शन हो गया उसका ट्रीटमेंट है राइट so this is what this person he monitored very serious people who recovered from disease they could nurse right. So the important part is that we have been in India. We are lucky that we get vaccinated naturally here. We blame the government, we get vaccinated. And let me tell you, it's not vaccine which work against only the disease. Vaccines are developed against addiction. The people who take drugs, you can develop vaccine against morphine, you can develop vaccine against heroin, right? So what do? You generate antibodies, you generate immunity, and now you're prevented. The same with pollution, allergens, you get allergy, and if you develop antibodies, you're protected from allergies. So it is important that our immunity should be primed, immunity should be boosted through vaccine as well as through natural infection, natural exposure as well. But let me tell you that before the dawn of vaccine, where the Western countries started working on vaccine, about 1500 AD, there were physicians from Turkey and China. They were practicing vaccination. They were practicing variolation. So important part was that these Turks and Chinese, they will allow their children to play with the children who are sick. They're going to send the children to जाओ खेलो उन बच्चों के साथ जिनको बीमारी है तो हम क्या करते हैं उन बच्चों के साथ में खेलो बीमार है because they realize if their child going to play with the children who are already suffering from disease, they will get exposure to that particular disease. At the same time, they may get mild infection because the quantum of infection not be that much, the magnitude may not be that much that will induce full-blown disease. So they allow the children to play with, the kids who are suffering from disease. But they also realize that especially in case of smallpox, so in smallpox, you get blister, you get pustules. So whatever was the secretion from these pustules, they used to extract that secretion. And this secretion, they used to attenuate, they will keep for some time so that the bacteria, viruses will lose their virulence. And this secretion they used to inject in people. So the people, they were immunized, or they had very mild sort of fever, and they recovered. And when they recovered, these people were not infected, right? These people, they were not infected with that particular disease. So this was regularly being practiced in Turkey and in China, right? So what happened that later on, there was one, ambassador from England, he visited Turkey. and he was in Istanbul. And this ambassador's wife, who was Lady Montenegro, she observed that the children, the healthy children, which were playing with the sick children, and ultimately these children will never develop disease. So what she did, she used to send her children as well to play with diseased children. And she realized that her children, they were protected from those diseases which were prevalent in Istanbul. Then she learned, she became very much intrigued. She learned from the physicians in Turkey the process of variolation. While she went back to London, to England, she started teaching about variolation. And this variolation, in no time, it became, quite popular throughout Europe. The people, they started, immunizing themselves through variolation. So why vaccine is important? The vaccine is important because the mankind has suffered, very large, tragedies, traumas through infection. For example, the smallpox, it killed 300 million people worldwide in 20th century alone. And the most of the native inhabitants of America, they were, wiped out. You see that the people they were infected with the Spanish flu. It was epidemic. 50 to 100 million people died because of Spanish flu. And then Black Death, which is caused by Yersinia pestis. From 1340 to 1771, 200 million people they died because of this disease. Typhus killed about 3 million people between 1918 to 1922. And this is a very interesting story of China in Italy. So this cathedral, it was, constructed. And what happened then in this Siena, there was a flood in 1348. And the flood, it wiped off at least 70% of the population, and including the people who were building this cathedral. And what happened, that ultimately, in the memory of those people, they left this cathedral as such. So the flood is wiped off, about 70%. So, nearly majority of the population of the city, it used to be wiped off from such diseases. In India, we had a lot of epidemics, many epidemics of cholera. We suffered about 8 epidemics, and thousands of people, they were killed. Malaria has killed from 1600 up to 2 million people. They killed 2 million people each year because of malaria. AIDS from 1981 to 2016 has killed 35 million people worldwide. Okay, and now we have all experienced this devastating disease, which is which was infected by coronavirus. And we had, the coronavirus, it infected about 60, 70 crore people worldwide. There's 70 crore cases. And there are about, 70 lakhs dead because of coronavirus. So why that is urgent? What does urgent mean? Because if you get infection, you can treat. Or is there any remedial measure before you get treatment, before you get infected? You feel like you need accident, why don't you put helmet? Or you purchase a proper car with at least 6 airbags in it. So the important part is, how does vaccine work? So what happens is, I told you, the immune system recognizes vaccine agents as foreign, destroys them before they cause disease, right? And remembers it forever. So what does it mean? There are 2 important parts in the vaccine. The first is, anybody who claims about vaccine, the vaccine should protect from the disease. Second important cardinal feature of vaccine is that vaccine should induce long-lasting protection. If the vaccine is not protecting you for a long duration, then it is not a vaccine. It's something like immunomodulator. Right? The cardinal feature of vaccine, for successful vaccine, is that it should protect, the same time it should induce long-lasting immunity. People who have been vaccinated against, for example, polio. There was a lady in UK, she was vaccinated with polio, and she was 85 years old. And she still retained immunity against polio. So her cells were taken and they were exposed to polio virus antigens, and her cells reacted. Indicating that whatever polio vaccine was developed, smallpox vaccine was developed, they induce enduring memory, long-lasting memory. So this is one of the important factors about the vaccine. When the pathogen infects, can you please mute your mic? The body recognizes a response by neutralizing the target antigen of the pathogen. It multiplies, and then ultimately we are prevented from the disease. Vaccines, as I told you, these are the most effective remedial measure of 20th century. So we have got through vaccines, there are a lot of victories which have been achieved. Right? And these are not ordinary victories. That some people are treated, some people develop drug resistance. The biggest advantage of vaccine over drugs is that when you treat with drugs, so people, besides the people they are cured of drugs, at the same time, the drug, they're also responsible for generating drug resistance. Okay, so many people, if they become drug resistant, for example, in case of HIV/AIDS, for example, in case of tuberculosis, many diseases, a person develops multiple drug resistance, or now we say when they are developing extremely drug resistance microorganisms. So they are not treated with any drug. So multiple drug treatment is given, and these people don't respond. So as you can see, in case of tuberculosis, somebody gets XDR is like a death sentence. The person gets like a death sentence. But in case of vaccine, if it's a successful vaccine, you're going to develop immunity. You're not going to develop any drug resistance. And the advantage of vaccine is that you only get just one shot, or at the most 2 shots, that's enough. But for drugs, you have to keep on treating the person. Every time you have to treat the person first. Right? It is the cheapest form of prevention or even elimination of diseases. So that's why I say that vaccine can conquer diseases. So victory of vaccines. So why the victory of vaccine? This slide is evident. But if you see here, in this slide, that this is a data of 2021. So how many diseases the vaccine has eliminated from this earth? If you see here, smallpox. Many of us, like when I was growing up as a child, I still remember that whosoever suffered from smallpox that time was a social stigma. So there was smallpox used to infect the body of a patient and the mind of public. So it became a social stigma. So people used to not only get disease, but this was a debilitating disease. It used to inflict a scar on the face. So once the person used to get smallpox, it's a whole life. He's going to carry a scar on the face, on the entire body, and the people remember, oh, he suffered from smallpox. So some people said that he's a sinner. He committed sins. And that's why he got infected by smallpox. It was a debilitating disease. It scars on your face. Right. But now you see polio, it is totally eliminated. 100% elimination. And there is no single case now seen in the whole world reported for polio. Diphtheria, totally eliminated. Smallpox, rubella, mumps, polio. Polio, again, a very debilitating disease. It used to infect children. And the people, the children who used to get infected, their limbs used to be involved. And their legs were like paralyzed. Their hands were paralyzed. The bone was stopped flowing. And the child who suffered from polio, for them it became very difficult to even walk throughout their life. They cannot use their limbs throughout their life. So this is the victory which the scientists have, or the vaccine has achieved. Right, pertussis, totally 99%, measles 99% elimination. Okay, so these are some of the cases. In case of measles, only 9 cases reported in 2021, and pertussis 1609. Right. So many diseases, this is not a total figure, many diseases have been eliminated because of vaccines. Okay, so if you see here this graph, this is the number of people who suffered in thousands, and this is the year, and this shows about the polio and about measles. So if you see here graph, there was total after vaccination, it was total dip in the cases, and almost this has reached zero, in fact, 0 cases. And same in the case of measles. Right, so besides, I told about polio and measles, even Haemophilus influenza type B, or which is HIP. So when vaccine was introduced in 1988, 1 in 200 children aged less than 5 years they developed HIP. And these 15 to 30% of infected children developed hearing impairment. They lost their hearing. Okay. So this again, HIP was a very, it was a debilitating disease. So ultimately, when vaccine developed, many of these cases quite effective, and very rarely we see HIP cases. Rubella. So 1964 to 1965, 20,000 infants were born with deafness, blindness, heart disease, mental retardation, and other birth defects. And after the vaccination, there is no threat. So the message over here is that it's not only severe sickness, it's only not that you get fever and then you recover after lying on bed for 3 days. But you can see that how much they loss these vaccines have inflicted in humankind. Okay, so it's very clear now. If you don't get vaccine, this is the situation you're going to have. If you get vaccine, you're a healthy person, right? So protect your children, immunize for good, and should not forget about immunization. And what are the attributes of an ideal vaccine? So I told you that it should generate the right type of immunity. While you're working on vaccine, you should see, first of all, important part is you should understand that what is the immunity which the host requires to protect against that particular organism. That is, host-pathogen interaction should be clear. The vaccine to protect loss-lasting immunity should generate adaptable to mass vaccination. So there may be many vaccines, but the vaccines, they are not adaptable to mass vaccination because you can produce those vaccines. You have to immunize an entire population on the earth. So it's also important that there should be mass production. It's stable with long shelf life. So vaccine should be stable. It should not be that you develop in your laboratory or in the company after it reaches for immunization. So the vaccine loses its potency. It loses its efficacy. So it should be effective, long shelf life, safe with minimum side effects. So vaccine should have minimum side effects. It should not be that it may induce some side effects. The side effects may be sometimes more dangerous than the beneficial effects. So many vaccines they don't pass because of the side effects. Cost effectiveness. So vaccine should be cost effective. Yeah, it should be easily affordable by everybody. And then acceptability. So vaccine, it should be acceptable to all people, right? So there should not be any component, the person, many beliefs or, the background of the population should be taken into consideration while developing vaccine, right? So vaccine should not have any such product which people will not accept. Okay, so that is also very important. So there are many types of vaccines. Initially, like vaccines were started by Louis Pasteur, Edward Jenner. So these are the people, Louis Pasteur and Edward Jenner. They are considered to be pioneers. They show the dawn into the vaccine research. So they started with vaccines which were live vaccines. The vaccine was injected, and these vaccines they ultimately going to induce a little bit of infection after the person recovers, and then it induces immunity, right? But if you remember Louis Pasteur experiments that he would develop a vaccine against anthrax, and when he used to inject anthrax into chickens, they used to die. So while he was culturing anthrax, until the years, what he did, he went for some vacations. And when he came back, he saw that this anthrax, whatever medium he was growing in, it got evaporated. And it was only powder. So what he did, he again, used that anthrax. And now he immunized these chickens. And to his surprise, chickens, they had very mild form of fever, and they survived. So what he realized that probably with the change in the growing condition, or there was no media, so this microorganism, the bacteria, it has lost its virulence, or it has become attenuated. It is not dead, but it is attenuated. And when he, after these anthrax bacteria injected, then he inoculated fresh anthrax. And again, to his surprise, that these chickens, they survived. So this is how he developed the concept of a vaccine. So people then started using killed vaccine. That is, they take microorganisms, bacteria, viruses, kill them, and they also work. Then subunit vaccines. But many of these vaccines, the microorganisms, they did protect it, but some of them, they also had a lot of side effects. So they started, identifying the beneficial portions or the immunodominant antigens of these microorganisms. So immunodominant antigen was used, and remaining antigens, which were suppressive, which induced autoimmunity, were discarded. So this is how subunit vaccine they started. And with the advent of recombinant vaccines or recombinant technology, then recombinant vaccines were produced, because when we produce subunit vaccine, we need a lot of protein. And the protein extraction of protein became very difficult. You have to grow bacteria, and then you have to purify protein. But with recombinant technology, the genes were cloned, protein was expressed, and now it was easy to produce protein in large scale. Then people realized that in protein, again, with many portions, these portions of the epitopes, these are protected. Some epitopes, they are autoreactive, they can induce, and some can suppress immunity. So they identify protected epitopes. Then DNA vaccine. So whole DNA was taken as a vaccine, which is immunized, and the people got protected because of the protein secreted. Yeah. Then animal vaccine. So it became very difficult. Many countries, like African countries, many Asian countries, it was difficult for them to afford seeding and needles. So the proteins or the antigens, they were expressed in banana, in tomato, in potato. And this is how the edible vaccines were produced. And most recently, the mRNA vaccine. So this is what the mRNA vaccines has to be developed because of the creation of COVID-19. So COVID-19, as I told you, that 19, that is, it came in 2019. And till now, it has infected nearly 70 people, killed about 70 lakh people, and it posed a big threat and challenge for scientific community, right, to entire world and challenge to scientific communities. Many people, they started working on COVID-19, and these people, they were already working on mRNA vaccines, both Kathleen Perico and Drew Weissman. So Kathleen, she's a biochemist, and along with Drew Weissman, they were interacting, and they were working on mRNA vaccine. So these people, they have, before even COVID-19, they have made a significant contribution on mRNA vaccine. And ultimately, their work laid the foundation for developing a vaccine. So there was a breakthrough against COVID-19, and they joined hands with companies like Pfizer, BioNTech, and Moderna. So let me tell you that in many universities, many institutes, people have a lot of potential, and they develop many important things that can be beneficial for they have a lot of societal impact, societal benefit. But see, the scientists, they don't have capability to take ahead their product to market or to use it for further for public benefit. So this, whatever research they do, it may have a lot of potential, but it remains up to their publications. Okay, so the only benefit they get publication. I was attending one conference in South India, Karapudi, and there was one person from US, who is a professor. So he came especially, he said that especially I came to this conference, a small conference, and I came over here just to thank the person to whose technology, to whose paper I developed the entire company, right? And I was so much surprised that it has so much of potential in his work. And this person only published a paper in Indian journal. I mean, not even taking a patent. So I looked onto it. I developed entire, further into this concept. And now my company is of this many $1 million. Especially I came to thank this person and personally meet this person. So many of us, we have so much of credibility, so much of potential. And even in this lecture hall, there are many scientists, like Dr. Prince, okay, and many other Mishima and Dr. Mrs. Prince's. So all of them, many people, they have a lot of potential. So they don't, they require hand-holding with companies. And I remember we once published in 2002 one paper, and then in 3 years, Biogen company developed technology, humanized antibody on it, and 3 years they took it ahead for clinical trials, started treating patients suffering from relapse and refractory follicular lymphoma. And the company took 6 patents on it. I had a little idea that we may have potential to treat cancer. And even our patent people, the people who are expert in every university, every institute, and in fact that even CSI has, patent institute. When I sent for it, it said, oh, it has already got so many patents, and probably this was, nothing, there's no novelty in it. we may not get patent on it. But in spite of that, there was a novelty. There were many patents on that work, but Biogen did 6 patents. Right. So hand holding is very important with the companies. That is very important. So what is mRNA technology is and why it brought a breakthrough in it? So both of these professors, they were focusing on like mRNA-based therapies before it reached to development of COVID-19 vaccine. So we all know that the body system can respond to synthetic RNA. So people that developed synthetic RNA and they were trying to use synthetic RNA for therapy. But somehow synthetic RNA therapy is not effective, it's ineffective. So what they did, they tried to modify the chemical structure of synthetic mRNA. And the reason was that this synthetic mRNA, if you inject, if you inject a synthetic mRNA, so besides generating protein, ultimately RNA is going to produce the protein. So they were inducing like polyclonal activation of B cell T cells, and a lot of pro-inflammatory cytokines they were getting secreted out. So here was the breakthrough and novelty. What they did, they replaced uridine of RNA with a similar molecule, which is called as pseudo-uridine. And when they replace this uridine with pseudo-uridine, they reduce the inflammatory response triggered by mRNA. Right? And this inflammatory response was highly dominated over the response generated by the protein. Right? So immune system was not activated against the protein of interest, the protein which ultimately going to protect you as a vaccine. Okay, so this modification was safe, it was quite effective, and could be used for mRNA therapies. And ultimately, they used it for vaccines. So this is uridine, and this is pseudo-uridine, which they produce. So the difference in pseudo-uridine is that at P5 of glycoside isomer, this is isomer of this, of uridine, and it contains CC carbon-carbon bonding between C1 of the ribose sugar and C5 of uracil, rather than usual C1 and N1 bond found in the uridine. So this change, ultimately what they did, when they did experiment, they observed that this unmodified mRNA, it since induced a lot of inflammatory responses, and when they injected modified, it was quite interesting that this mRNA, it did produce a lot of beta protein, high quality protein, whereas this synthetic mRNA didn't produce much protein, and the response was dominated because of the inflammatory response, whereas this ultimately suppressed inflammatory response and led to generation of high immunity. So it was a very robust immune response. So ultimately, this with the appearance of COVID-19, they utilize this technology. So in case of synthetic mRNA, when they do modification, with this modification, they could very significantly increase not only the production of protein, but also the immunogenicity of protein. The immunogenicity of protein was also increased. So ultimately, they did collaboration with the Bionet, Moderna, and this Westman and Calico research, it ultimately caught the attention of many pharmaceutical companies. But ultimately, they signed an agreement with these companies, Bionet, Pfizer, Moderna, and this is how they entered into development of vaccine. So they took spike protein. So the virus, it is like a spike, it's like a crown. And in the crown, like you put different germs. So in the virus, there are all these spikes which come out. And through this spike, ultimately they attach the cells of the body, and then they infect. They infect the epithelial cells, they infect a lot of cells of the body. So the target protein they identified was the spike protein. If you can block the spike protein through antibody, the spike protein, it binds, it enters in the body. So if the body can produce antibody and a spike protein, then you want to neutralize this spike protein from entering, attaching to your body cells. You're preventing attaching to lung cells, and ultimately you'll be protected. Right. So ultimately, they transferred this technology to developing vaccine, and this technology ultimately it worked well. People were vaccinated, right? And after vaccination, the people they were protected. So the biggest advantage of this mRNA technology is that you can develop vaccine in a very fast mode, right? So this technology allowed for a remarkably rapid vaccine development. So we have all experienced that within like 2 years, the vaccine was available, right? So it also, the development of the vaccine, the progress of the vaccine is impeded because of the development as well. So development, as I said, the vaccine should be produced to an extent that it can be used for mass immunization. If you produce a vaccine that can immunize 10,000 people, but every day, 1 lakh people they get infected, but you have capability of producing only 10,000 doses. It can be effective. It is very effective, 100% protection, but only 10,000. So that is also a hurdle. But the development of mRNA technology, it overcame that hurdle, that the vaccine was very rapidly developed. So the ability to quickly adapt the mRNA sequence in response to a new variant of the virus is a significant advantage of this approach. So if there is even a mutation, there's a variation in the virus. So biggest problem or biggest challenge developing vaccine against viruses, because the virus it has a lot of mutation. So the biggest challenge for developing HIV vaccine is that the HIV/AIDS virus it mutates very fast. So a lot of antigens drift and shift. So it becomes a very challenge. You develop a vaccine against one particular strain, which is showing this particular antigen protected. After that, this virus, it mutates this protein, which is immunodominant. It brings up another protein. So you have to develop a vaccine against another strain. The advantage with mRNA is that you can develop quickly a vaccine against second strain, the third strain, fourth strain. Okay, so it had this ultimately technology has a great impact, it has a breakthrough technology. So ultimately, they went through vaccine trials, and then FDA approved, and they got authorization for development of vaccine, and in emergency, it was allowed to immunize people in many countries, right? So their success has also spurred further research and development of mRNA vaccine-based therapies for various diseases now. A lot of laboratories were working to develop vaccines against many diseases, right? Like diseases like HIV/AIDS, tuberculosis, cancer. If this technology may be used, definitely it will have positive impact in developing vaccines for public benefit, public health against many diseases for which vaccines were not developed yet, in spite of a lot of research and different type of vaccine technology have been used. For example, if you take an example of tuberculosis, 100 years back, BCG vaccine was developed. The development of a lot of technology, DNA technology, right? Some unit vaccine technology. But after 100 years, even we don't have vaccine against tuberculosis. So now, this approach is being used to develop vaccine against tuberculosis as well. So ultimately, overall view to see how this mRNA vaccine works. So this is SARS-CoV-2, the spike protein they have taken, and this spike protein they have generated mRNA, they develop a vaccine, and they have checked if you immunize in mice, or they have tried in non-human primates. In monkeys they tried, and this vaccine, mRNA vaccine, they injected. And this mRNA vaccine is ultimately taken up by cells, for example, by genetic cells. And these genetic cells, they process the protein, which is ultimately, this mRNA vaccine, it instructs a body cell to produce protein. So this protein is produced in the cytosol. It is taken up by, for processing. So it is processed. And immunodominant epitopes, they bind to MSC molecules. And these MSC molecules ultimately they present these epitopes of this protein, this spike protein, to T cells. And these T cells, ultimately they're going to proliferate. It is taken up by B cells. B cells, it processes this epitope, presents to T cells. T cell provides help to B cells. And these B cells ultimately start producing antibodies against this spike protein. So these antibodies, another advantage of mRNA vaccine is that as compared to native protein, the mRNA vaccine reduces production of high affinity antibodies. So you may have antibodies, but antibody may not neutralize virus. You can neutralize virus if you're producing high affinity antibodies. So this vaccine ultimately induces production of high affinity antibodies. And the advantage over here is that the B cell produce high affinity antibodies. These antibodies ultimately, they bind the virus, neutralizes efficacy, and then eliminate it. The advantage is that it induces the production of both CD4 T cell and CD8 T cells. Whereas CD4 T cells, they are responsible for providing help to B cells to produce antibodies. CD8 T cells, ultimately they survey, they patrol and identify the cells which are infected by virus. And the virus which are hiding inside the cells, they recognize and then ultimately they kill them. So this cytotoxic T cell is very interesting T cell. This cytotoxic T cell, whenever any cell, like antigen presenting cell, they come in contact with the cytotoxic T cell, or the infected cell, they come in contact with cytotoxic T cell, and they kiss this infected cell. And this kiss is a kiss of death. Right? It is like kissing a cobra. Anybody of you have kissed cobra? Okay, you can kiss and have experience how cytotoxic T cell is killed. Right? So ultimately, the hidden bacteria is eliminated and taken up by more immunocompetent cells, they phagocytose and then they crush it, eliminate it. So this is how this vaccine is working, producing antibodies, producing cytotoxic T cells. Cytotoxic T cell identify infected cells and it keeps on eliminating. Right. So what are the benefits of this vaccine over traditional vaccine? The first thing is that, for example, DNA vaccine, it always has fear of using DNA vaccine that it can get integrated into our genome. But mRNA cannot be integrated into our genome, so there is no potential danger of insertion of mutagenesis. mRNA only requires cytosolic delivery in the host cells, where it is translated into functional protein. That when protein is produced in cytosol, so you're leading to production of CTLs, cytotoxic T cells. To produce cytotoxic T cell, you deliver antigen into cytosol. So antigen is delivered into cytosol. mRNA is translated transiently. This is also important. In cells and is degraded in a relatively short but controllable amount of time. So it does not remain in your body, so keep on secreting, protein. It's not so. So it's translated transiently into the cell. After that, once immune system is activated, so it is degraded. So no need for culturing pathogens here. You're not using pathogen, you're not using live pathogens, or always here. If you're using live pathogen and it is not attenuated properly, it can kill. And there have been incidents where children have been killed by live vaccines. So it was not properly attenuated. So it generates strong immune response. Scalability production is easy, right? And in future, mRNA vaccine can be used for personalized vaccination. Some people may be, protein may be immunogenic for you, but it may not be immunogenic for me. So based on understanding person's genomics, and genetics, you can generate vaccine, right? And in using the most of the vaccines, let me tell you, 99% of the vaccines are failed because they cannot induce long-lasting immunity. So FDA will not allow. In last 5 years, 44 vaccines were developed against HIV, and all of them were failed because they could not generate very beneficial, robust immunity, but they failed to generate long-lasting immunity. Right? So you can generate long-lasting immunity, very strong memory, and B cell and T cells. Long-lasting protection. So you need only one vaccine or two vaccines. Right, but there are some disadvantages always, but the disadvantages of mRNA vaccine are quite less as compared to their benefits. Right, so this already I have told you, and now currently after the success of coronavirus vaccine, many groups, many companies now they are working on using mRNA vaccine for cancer treatment, other virus treatment, very rare diseases, autoimmune diseases, like protein replacement therapy as well, neurological disorders, infectious disease, regenerative medicine, monoclonal antibody production. Right. So it has a lot of implication now. And finally, I would like to thank you all and dedicate this lecture to both of the Nobel laureates and open for now questions.
Questions about the longevity of the virus if we know not doesn't have to so what is your take on that?
As far as that was a big question during immunization with this vaccine. Many people had a lot of concern because some people, there were a lot of non-specific comments on the major comment was that the vaccine will take a lot of time, 10 years, 20 years, to declare whether it's fit for human use or not, is this vaccine or not. For example, BCG. Once upon a time, it was thought that BCG should be stopped in India because when the clinical, when a study was done, and through that study, they said that BCG does not work because there was regular increase in TB patients. So to prove that, there was a project which was funded, and the study was taken for 10 years. After 10 years, they concluded that BCG does not work. But this, within 1 year or 2 years, the vaccine is now ready. But that was emergency. You have to now check that what do you want? Do you want death or do you want to take risk? Same way, when this pesticides were invented and people, it was a big problem for crops because crops being destroyed by different insects or viral infection, bacterial infection. Ultimately, there was a suggestion that we should use pesticides, but there was resistance. the pesticides they can cause cancer. Ultimately, it was decided about a lot of debate. What do you want? Do you want death of your family because of hunger? Right? And that is 100% death. If you don't get food to eat, it's 100% death. Or do you want to die because of cancer? But that is not, everybody is going to die because of using pesticides. So out of 1 lakh, 1% will develop cancer. But if you don't have food, every person is going to die. So ultimately, when it was, weighed, so the amount of tragedy the person going to be inflicted by hunger was much more as compared to use of pesticides. So ultimately, pesticides are recommended. The same thing when people in scores of people dying because of coronavirus. What to do? It was ultimately about emergency. And now your question whether it induces long-lasting immunity. So in case of immunological studies, it has been seen that if your T cells and B cells that develop memory cells beyond 45 days, so usually they remain for long periods. So based on those experiments in mice and in non-human primates, like monkeys, it was found that it was inducing memory T cells and B cells. So that is the reason it was allowed. So some of the data which is now published, it's coming up, it's showing that memory generation is a problem. Memory generation usually, like if you get 1 vaccine or 2 vaccines, you should develop immunity for your whole life. So that is still under consideration. But even if it was working like immunomodulator, the people got sick, animals showed protection against challenge with coronavirus. So that is the reason it was allowed. But short-term memory, yes, it induces short-term memory. And that was the reason, 2 shots or 3 shots were given. The long-term immunity still, data has to be produced. Yes, a lot of the foundation studies being done, like trying these with mRNA and protein vaccine. But still, as far as your question is concerned, it requires long-term studies to firmly establish that the acid is inducing long-lasting immunity. Self-flying is a problem. But that again, you can use these smoke refrigerators or ice packs, and it worked well. So storage or the cold chain was not a problem during coronavirus pandemic. So the students are very good. They are trained to answer questions, not trained to ask questions. Or they respect teachers, they don't ask questions training the teacher. Or they're not allowed to ask questions. I just want lecture. Do you think any lecture is a career is a lifelong human? Or do you think there is a chance of real? See, that's a really important question. And that's why before a person is vaccinated, the vaccine will undergo extensive clinical trials, extensive checks. And FDA is very critical on it. Many vaccines are produced, but if they protect but don't induce long-lasting immunity, they don't allow. Especially in certain diseases which are not, exotic diseases or not even create emergency. The coronavirus created emergency. Many people laxative were dying, living their life. So they're not going to allow for tuberculosis. There is an alternative drugs are there to treat. Yes, but whatever vaccines available, they are passed through many clinical checks. I give you an example of vaccine that a lady was immunized after 85 years and they tested she was responding. The same way like polio, smallpox vaccine, measles, they generate long-lasting immunity. Whole life person has developed immunity. So what about the students? Can I ask questions to you? I think. Yes, वो लड़की डर गई. ये follow ना करो. Yes. You can introduce yourself. BSc? BSc Biotechnology. Microbiology. See, in your class you may have many friends, right? Some friends you will just say hello, and some friends you will really meet. But there will be some friends you have a lot of intimacy. You want to meet every time. So even if you have high affinity, right? So high liking for that person, so this is high affinity. You're not meeting other people, meeting them rarely, or meeting them during lunch or dinner. So that is low affinity. You don't meet any people, that is, you're not developing any affinity. You're not developing antibody. The same way, when we have B cells, when there are different type of B cells, and it depends on what condition B cell is primed. The B cells is going to produce antibodies, and these antibodies are of high affinity. So these high affinity antibodies, they're going to very strongly react with the bacteria. Right? It's going to bind. Association rate will be fast with this bacteria, and dissociation rate will be very small, very slow. Once it has caught, so ultimately these antibodies are really going to adhere to it till they take this virus or bacteria to its graveyard, to destroy. So either the high affinity antibodies, they're going to bind, complement, going to work, and it's going to kill it. Or high affinity antibodies, they're going to take it to macrophages, bind the Fc receptor, and it will survive, kill it. So low affinity antibodies, these antibodies are also against the virus, right? But they're going to bind slowly and maybe dissociate as well. Bind, dissociated. It may not give ample time, may not have sufficient affinity, not sufficient ample time, so that they can bind to an extent that complement works and destroys, or they take it to macrophages, utilize, and they kill. So this is the difference between high affinity antibody and low affinity antibody. The role of mRNA is that mRNA, it is producing for generation of high affinity antibody. The B cell should be exposed to sufficient quantity of antibody, right? So mRNA, since it is producing protein, right, so sufficient amount of protein is produced and sufficient amount of protein is coming on the surface of the cells the B cell can have enough optimal level of protein so that they get optimally activated so that they produce high activity antibody. So when you inject protein itself, so this protein now it gets dispersed in the body. And B cells, some of the B cells may get sufficient exposure of sufficient antigen if they are present nearby. How the antigen reaches B cells. If not using sufficient to B cell, you will not be activated. So this is a quantitative distribution. How many cycles of immunization will be required in future? For instance, whatever if we have been able to generate immunity, whether it's a small part or whatever, do you think there is a chance that this disease, the virus will come back? And if so, how do you detect? Okay, first question is how many immunization do we need? See, as far as mRNA is concerned, probably we may need 2 immunization. The reason is that is not like DNA vaccine. This mRNA is going to produce transiently and then the protein going to disappear. You need second booster so that your B cell and T cells they develop secondary response and develop into memory cells. So if this can induce memory B cell and T cell long lasting, enduring T cell and B cell, probably you need only 2 immunization. That's it. As far as polio is concerned, especially like virus, we are using polio vaccine. The virus they give is attenuated form, it keep on dividing or it keep on releasing antigens. So the cells get sustained release, a sustained source of antigen. So then the immunity or the memory cells which are generated, they keep on getting exposed and ultimately they generate long-lasting immunity. It's very simple, like, same way, for the first time when, I meet you, we're going to introduce ourselves, right? And then we keep on meeting very frequently. Every evening we are meeting, drinking tea in Punjab University, right? After some time, 6 or 7 months, I can realize, even if you're walking about a kilometer ahead of me, I can say, Professor is going there. Not because of constant exposure with you have developed in memory. The same is for T cell and B cells. The constant exposure of antigens, they have been able to develop long-lasting immunity. So exposure of antigen is very crucial. Whereas in case of replicating viruses or live attenuated vaccines, like polio measles, in that case, the constant antigen exposure is available. But at the same time, once these cells are generated, especially whether these are T cells, they are such cell in body, they remain for whole life. They can live even longer than us, right? So in that case, they're going to generate response. And once they develop memory cells, as even if you're walking a kilometer ahead of me, they respond, I'm going to respond rapidly with mine. And same with now they respond very rapidly. They respond to very low dose of antigen. Even low dose of antigen is detected, they don't respond fast and very rapidly. Any question online? People available online? No question? Okay. So shall I start asking questions now? Okay. Okay, thank you very much. It was an excellent one. Hmm.